This video looks at digestion and absorption and is for A-level biology. Have you ever wondered what happens inside your body to a burger you've just eaten? Or why some people can't drink milk and are lactose intolerant? And what does lactose intolerant even mean? This all happens by a process called digestion and absorption, and we're going to look at these two processes in more detail. If we start with digestion, digestion is the breakdown of food from large insoluble molecules into small soluble molecules. And absorption is these small soluble molecules from digestion being taken into the bloodstream, so they can be utilized essentially by the body. If we break down digestion, it involves two main processes. The first is mechanical digestion, which is where teeth and muscular contraction break down these large pieces of food into smaller pieces. This is really important as it increases the surface area for the action of many different enzymes. Next, we move to chemical digestion, and this is done by enzymes. And they're able to digest a wide range of different biochemicals or macronutrients, such as protein, carbohydrate, and fat. And importantly, each enzyme has its own optimum temperature and pH, which means different enzymes are found at different regions in the body. I'm going to look a bit more about this. So if we look at the mouth has a kind of neutral pH, varies a bit, but it ranges from, say, 6.5 to 7.5. And one of the key enzymes here is salivary amylase, which is involved in the breakdown of carbohydrate. If we move to the stomach has a more acidic pH, and that's due to the presence of hydrochloric acid. One of the enzymes in the stomach is pepsin, and that helps the breakdown of protein. Next, we move into the small intestine and we look at the duodenum, so the first part, and that has a higher pH. And this is due to the alkaline fluid that's being secreted from the Brunus gland in the submucosa of the duodenum. So it's 7 to 8.5. Lots of enzymes are present here from the pancreatic juice, trypsin, lipase, and pancreatic amylase. If we move on to the ileum, so next part of the small intestine, this is a slightly more acidic pH, and the enzymes here are disaccharidases. It has a slightly lower, but more acidic pH here due to the presence of fatty acids, which make it more acidic. The colon has the same, roughly the same pH as the ileum. So now if we take this burger and we think about the fact that enzymes are present at different locations in the body because they have a different optimum pH, we're going to look to see how this burger, so how carbohydrate, fat, and protein are broken down in the different regions of the body and by different enzymes. So if we look at carbohydrates first, carbohydrates are broken down, so polysaccharides are many monosaccharides, so those hexagon squares, joined by glycosidic bonds. And if you take the example of starch, this is broken down into its disaccharide, maltose. And disaccharide just means two, so two monosaccharides joined by a glycosidic bond. Water is involved in this reaction. It's a hydrolysis reaction, and that's why there's a water symbol there. This takes place in both the mouth with salivary amylase and in the small intestine, pancreatic amylase. The next stage of carbohydrate digestion is we're now taking this disaccharide and we're breaking it down into a monosaccharide. And this happens in the ileum, epithelial cells in the ileum. So you can see the three examples of disaccharides. Maltose gets broken down by maltase, the enzyme, into glucose and glucose. Sucrose gets broken down by sucrase, the enzyme, into glucose and fructose. Lactose gets broken down by lactase into glucose and galactose. And here we see that people that can't digest lactose properly don't have the lactase enzyme present here. If we look at this now, just sum it up in terms of where it happens in the body. In the mouth, we start with salivary amylase. We've got this polysaccharide being broken down into the disaccharides by amylase. This also happens in the duodenum. And then the next step is the disaccharide being broken down into the monosaccharide, which happens in the ileum. And we can see that here. Disaccharides are broken down into their monosaccharides. So next we're going to look at proteins, so how they're broken down and by what enzymes. Proteins are composed of amino acids all in a polypeptide chain. And this is just an illustration showing lots of different amino acids joined together. This is not a secondary or tertiary structure, and there is no folding at present, just a straight chain. There's two key enzymes you need to know about. And the first is exopeptidase, and these hydrolyze or break the peptide bonds between the amino acids at the ends of the polypeptide chains, so terminal peptide bonds. And you can see the arrows pointing here. This continues, so we can see breaking the ends, so the terminal peptide bonds. And you can see keeps breaking it until it gets much smaller. The problem, though, is if there's a huge long chain of amino acids, really, really long, it's going to take a while for the exopeptidase, which breaks at the ends of the chain, to fully break up the amino acid. And that is why another enzyme is involved, an endopeptidase, endo being in the middle. And these hydrolyze the peptide bonds between the amino acids in the middle of the polypeptide chain. So you could say the non-terminal peptide bonds. You can see it breaks the chain here. Following the action of the endopeptidase, it's now a lot easier for the exopeptidase to hydrolyze the peptide bonds at the ends of the terminal chains. And this combined action of endo in the middle and exo at the end increases the rate of protein digestion. Now let's look at how they are involved at different regions of the body. So we start in the stomach. So remember, the stomach is quite acidic, pH, and we have pepsinogen, which is the enzyme precursor, and this is activated by the hydrochloric acid, which converts it to pepsin, and this helps to break down the protein. This is an endopeptidase, so it's breaking down the polypeptide chain in the middle. So you can see the arrow pointing to the middle. We now go to the small intestine, and there's another precursor. So trypsinogen is activated by enterokinase to trypsin, and this is another endopeptidase, so it's breaking it down in the middle. The reason why we have precursors is because we don't want the active enzyme to be present all the time, because this can cause problems such as digesting things that don't need to be digested. So next is the exopeptidase, and this also happens in the small intestine, and this breaks down, remember, at the end, so it breaks down into amino acids or dipeptides. So the combined action of both endopeptidase first and exopeptidase helps to break down the polypeptide chains. If we now move on to fats, we'll see how they're broken down. So in fats, there's a really important substance which is involved in the digestion, and this is bile. And this emulsifies the lipids by breaking large globules of fat into smaller droplets. And this is really important as this increases the surface area for the action of lipase. And lipase is the enzyme that breaks down fat. It also enables the small, very small droplets of fat to be absorbed directly into the bloodstream. Now, if we look at where bile is produced in the body, bile is produced in the liver, but it's stored in the gallbladder. And it's delivered to the small intestine, where it has its action of emulsification via the bile duct. And gallstones are very small stones. They're usually made of cholesterol that form in the gallbladder. And gallstones can block the tubes through which the bile flows from your gallbladder into your small intestine. So it's just important to note that some people have gallstones. They can cause quite a few problems, often treatable though, and they could potentially block the passage of bile to the small intestine. So, as we've said, lipids are broken down into small lipid droplets by bile. This then increases the surface area for lipase to act on them. And lipase breaks down these small lipid droplets into monoglycerides and fatty acids. These monoglycerides and fatty acids stick together to form these tiny structures called micelles. You can see here. And this all happens, as we said, in the small intestine. So we now move on to absorption. And digested products are absorbed across the ileum, so the ileum in the small intestine, across the epithelium. And the ileum are covered in these villi, which are adapted to maximize the absorption. And on the villi, on the epithelium, so around the outside, they have these microvilli, which increase the surface area, again, to maximize the absorption. They also have a rich capillary network, and this is to absorb and remove the products of digestion, such as sugars and amino acids, and it helps to maintain a concentration gradient. It also has lacteals, so lacteals help to absorb the products of fat digestion. They're very thin, and this is to reduce the diffusion distance, again speeding up or maximizing absorption rate. So now we're going to look at how amino acids from proteins, monosaccharides from carbohydrates are absorbed from the lumen of the ileum into the bloodstream. So they're both absorbed by a process of co-transport, which involves sodium ions. And sodium moves down a concentration gradient, i.e., because there are more sodium ions in the lumen of the ileum than there are in the epithelial cell of the villi. Whereas the substrate, whereas the amino acid, shown by the green circle, or it could be the monosaccharide, moves against a concentration gradient. And that's why this process requires ATP. So once a substrate, the amino acid or the monosaccharide, as shown by the green circle, has moved into the epithelial cell of the villi, it now needs to get into the bloodstream. But another thing that's important to know is that a sodium-potassium pump helps to maintain this sodium concentration. And that's really important, as it means that the substrate, so the amino acid or the monosaccharide, can diffuse, can go via co-transport from the lumen of the ileum into the epithelial cell. So this sodium-potassium pump is pumping sodium out of the epithelial cell and pumping potassium in. Next, the amino acid or the monosaccharide moves via facilitated diffusion because there's a higher concentration in the epithelial cell of the villi than there is in the bloodstream to the bloodstream, and then it can travel around the body via the capillary network. If we compare the process of amino acid and monosaccharide absorption to fats, it has a slightly different process. So as we said before, micelles are essentially monosaccharides and fatty acids, and they are constantly breaking up and reforming, and they can release monosaccharides and fatty acids, allowing them to be absorbed across the epithelial cell of the villi. Monosaccharides and fatty acids are lipid soluble. They're non-polar, and they can simply diffuse across the epithelial cell of the villi. So it's quite a simple first process. These are then modified back into triglycerides, and this process happens in cell structures such as the Golgi body or the endoplasmic reticulum, and this all happens in the epithelial cell of the villi. They are next modified to chylomicron, which essentially is adding other molecules such as cholesterol or protein. And these chylomicron can then be transported to lacteals, which we looked at previously, and carried around by the lymph system, which eventually drains to the capillary network. So we've talked through the digestion of carbohydrates, proteins, and fats, and how they are absorbed into the bloodstream.